most of chapter 1 is fairly straightforward in microbiology, and I'm not going to give you a video where I read definitions to you, so make sure you either use the PowerPoints that you have access to or you use the textbook so that you can fill in those definitions and learn the terms. The videos are really just going to get you through some of the tougher concepts in a chapter, and really the only thing that students tend to struggle with in chapter 1 is the taxonomy portion of the notes, and so I wanted to run you through some of that material just a little bit. So let's say that you were traipsing around in the woods and you found some organism that nobody had ever identified before. Taxonomy would be you essentially defining what that organism is. You would look at what kind of cells does it have? Is it multicellular or unicellular? And you would just go through this sort of list of definitions so that you could classify it based on your description. Since you were the first person to ever identify it, you could even name it. Binomial nomenclature is the process of naming an organism with a scientific name, usually using Greek or Latin, although we've gotten a little bit more lax in terms of how that happens. Classification is, once you do your description of that, you have to put it into all of the different taxa or groups that are listed in the what's known as taxonomic hierarchy. This is the current taxonomic hierarchy. It didn't start out this way though. And so a big chunk of this chapter is understanding where it started versus where it is now. So Linneaus is one of the first people who started to actually classify organisms and put them into groups. He had a bunch of different groups, but he only had 3 kingdoms. Now, coming back to this picture from just a second ago, now we have domains, but when Linneaus did it, the kingdom was the highest grouping that you could have within living organisms, and he only had 3 kingdoms. He had plants, animals, and then minerals. Minerals are actually not living things, and so we're not even going to talk about that, and in fact that kingdom went away fairly soon after he had invented it. He published his idea about taxonomy and classification in his little booklet, Systema Natura. It has turned into a quite large book at this point, but it started out as just a fairly small booklet. Now, Linnaeus, just an FYI, not a super awesome human being, but he did advance science in some really wonderful ways, and so that's why we're still going to be talking about him very briefly. So the groups that he had, the kingdom was up at the top. It is what's known as the most inclusive grouping or the broadest grouping, and again, every living thing that they knew about was put into one of those 3 kingdoms that he had, plant, animal, or mineral. And really, again, minerals were not living things. So in terms of living things, you could be a plant or an animal, and those were the only 2 options. Phyla are smaller groups. Phyla is the plural form of phylum. Classes are even smaller, and then we keep getting smaller until we end up with species. A species is the most specific grouping. There's only one type of organism that fits into a particular species. Like dogs are a species, cats are a different species, humans are a different species. Another way to look at this taxonomic hierarchy is that everything is a group of related things below it. So a genus is a group of related species, a family is a group of related genera, an order is a group of related families, a class is a group of related orders, a phylum is a group of related classes, and a kingdom is a group of related phyla. So this is what Linnaeus had come up with, which we still use this system today. So it's been in place for hundreds of years at this point. Now, as we started to develop more knowledge and better microscopes and more biochemical tests that we could run, we wanted to try to make sure that when we were grouping things, they were actually related to each other. They didn't just look alike. So a phylogeny is a group of organisms that are related. You can look at a phylogenetic tree to help you understand phylogenies a little bit. What a phylogenetic tree tries to show you is who's more related to who. So as you look at this one, what you can see is starfish are more closely related to brittle stars than they are to octopus and squid off over here, because these guys are on a different branch from these guys. So the things that share a closer branch are more closely related to each other versus things that have far apart branches are more distantly related to each other. When we first started to do phylogenies, we used observable features like, hey, everything here, it has spiny skin. Everything here, they have a mantle and some stuff that you guys wouldn't be all that familiar with. Everything here, jointed legs. As we started to get more advanced, though, we could start to use the actual DNA present in an organism to figure out who is more related to who. So this is one of the first phylogenetic trees that was ever created. They called it the tree of life back in the day, and it's a very primitive form of what we had going on. If I remember right, this one was made by Haeckel. Haeckel, I'm not sure how you say his name. Haven't met him personally because he's long dead, but there you go. So he had 3 branches on his tree of life. Linnaeus only had 2 because remember, minerals weren't actually alive. And he put everything onto one of these 3 branches off over there. So that's a primitive version of this. Now, we're going to kind of skip Haeckel, aside from what I just gave you a second ago, and move on to Whittaker. The reason I'm skipping to Whittaker is the Whittaker system was used for a fairly long period of time in terms of phylogeny and taxonomy. In fact, there are a lot of high schools that still teach the Whittaker system, even though it's really should have gone out of style 30 years ago. The Whittaker system added the 5 kingdoms that most people were more familiar with. So we still had plants and animals. Protists Haeckel had brought in. Fungi is a new group. And then we have Monera as well. Now, the reason that we get new kingdoms, we have microscopes now. Those microscopes allow us to see organisms that Linnaeus couldn't see. That would be all of the kingdom Monera. They consist of very small single-celled organisms. The cells are what are known as prokaryotic, which means they have no nucleus. They're really super tiny. Everything else you can really see with the naked eye, although fungi, a lot of those are even microscopic, and you need a microscope to be able to identify them. So we added these kingdoms because we could suddenly see things that we hadn't been able to see before. Then, this one's really just a definition for you guys, so I'm going to kind of skip past that. Where we went after Whittaker is we started to learn more about DNA. DNA is what we pass down from one generation to the next, and DNA is what makes every living thing essentially unique. You have more similar DNA to your relatives than you do to species that are more distantly related to you. So when we started to look at DNA evidence, it gave us a more accurate phylogenetic tree. Now, Woese, I'm not sure how you say his name again. Unfortunately, he died back in 2012, so can't talk to him about it. Not that he would have ever answered any phone calls that I gave to him, but again, whatevs. He was an American microbiologist, and he used rRNA, which is ribosomal RNA. There are lots of kinds of RNA. The 3 that we're going to spend the most time talking about in this class are mRNA, tRNA, and rRNA. The rRNA is actually part of the ribosome, which is the protein-making machinery that exists within a cell, and it is highly conserved because if you mutate your rRNA and break it, the cell dies real fast. And so when I say highly conserved, I mean it doesn't change much over time. The mutations in it tend to die off, and so we don't see those. So when you start to look at rRNA analysis, you start to see these things that are more closely related to each other based on actual genetic information, not just, hey, this has hair and this has hair. We're looking at the genes to figure out how related things are. And when he made his phylogenetic tree using the rRNA analysis, what he discovered is there's 3 branches, kind of like what Haeckel had, except instead of plants, animals, and protists, there's bacteria that all share similar rRNA sequences. There's Archaeans, which all share similar rRNA sequences. And then there's the eukaryotes, which are the things that have more complicated cells. They have their own rRNA sequence. So there were 3 branches based on genetic analysis. So they're more related because their genes are more related. Then those kingdoms that Linnaeus had made, plants and animals, and that Haeckel and Whittaker then modified, they fall within these 3 domains of life. So the moral of the story here is we start out with the information that we have available to us. And when Linnaeus was doing his work, we didn't have microscopes. We didn't know about DNA. We literally did not know that it existed within the cells. We didn't even know that there was genetic information being passed down. Like how that happened was still being debated. As we started to get more evidence, back in the 1600s, we started to get microscopes. We could see things that we didn't know existed before. So we had to add new groups for that. And then we started to look at the DNA sequence from the 1960s on because we knew about them at that point. And so the reason why taxonomy has changed is because we've collected more evidence over time. It's very important that you understand science is trying to arrive at a truth based on the information that we have at any given time. And if we get better information, you have to be able to change your mind. That makes learning about this very difficult because when I was taught this stuff, I was taught the Whittaker system. And then I had to relearn a new taxonomic system because we got better evidence to support that. I can understand why it's frustrating because I've had to do it myself, but it's important that you are able to keep up with the evidence that's out there. Now, there is a complicating factor in terms of phylogeny. So as you read through this slide that's up here, we tend to think of genes and DNA like they get passed down from parents to offspring, and that's the only option. And unfortunately for us, that is not the only option. For example, if you get a viral infection, a lot of times that virus adds DNA into your cells. That's a type of horizontal gene transfer. Bacteria are also really good at swapping genes from one species to another species. That is what's known as horizontal gene transfer. Now, if we were trying to make a phylogenetic tree and we happened to pick a gene that was getting passed from one species to the next, it would make it look like those two species were related to each other, when really they just happened to live next to each other and they swapped some genes between them. So horizontal gene transfer makes everything much more complicated, and it also makes understanding bacterial relationships very complicated. The taxonomy of bacteria has been in flux for the last 30 years, and it really hasn't settled into a specific place yet. And so I'm not going to make you memorize a bunch of phylogeny when it comes to bacteria because it's ever changing, because our understanding of who's related to who changes based on the new evidence that we have. And unfortunately, horizontal gene transfer just makes that more complicated. Okay. The last thing, well, second to last thing that I basically want to mention for you guys, I've got a little blurb about size that's in there, and it's very important that you understand some basic stuff. So usually in either A&P or freshman bio, one of the first things you do with a microscope is a cheek smear slide. You stain some of the cells that line your mouth, and you can see the cells, and they're nice and big, and you can see them even on low power. What I want to point out to you, though, is bacteria are much smaller than human cells. We have eukaryotic cells, bacteria have prokaryotic cells, and they are much smaller. That means they're going to be a lot harder for you to find on our microscopes, and you're going to have to take it up to powers that you weren't even allowed to use in A&P or in some cases in freshman biology. Just keep that in mind as you run through there. Now, there's advantages to being smaller. It gives you what is known as a high surface area to volume ratio. That means you can exchange materials between you and the environment a lot more easily than something that has a larger cell. There's advantages to having larger cells and being multicellular too, but that surface area to volume thing is something that's going to pop up periodically. The other thing that I want to show you in here is viruses are much smaller than bacteria. In fact, viruses are so small that you will not be able to see viruses with the microscopes that we have. Now, if you go to a lab that has electron microscopes, you can see them. But using a standard light microscope, you can't see, for example, the coronavirus under there. And so the size matters because how we're able to see them depends on microscopes that have resolving powers that may not be able to see some of these smaller things like they could see some of the larger things up there. Final thing that I wanted to show you, this is a reproduction essentially of Woese's phylogenetic tree that he created. And what I want to point out to you is bacteria and Archaeans back in the Whitaker system, they were both put into the kingdom Monera. And what I want to show you is they are not related to each other. Archaeans are on their own separate branch that's actually more closely related to the eukaryotes than it is to the bacteria off over here. So again, that's one of the reasons why we're moving away from the old Whitaker system where they had both of these in one kingdom. They don't belong together. They're not what's known as monophyletic. They're not related, so they should not be in the same kingdom. They are two completely separate domains because they're not related to each other. All right, last little blurb. The clinical focus for this chapter dives you much deeper and harder into stuff than I would expect somebody in chapter 1 to be able to figure out. They're giving you this case of a person who presents with symptoms that are like meningitis, which meningitis is relatively common. But then it takes this abrupt turn and it starts to talk about prions, which chapter 1 doesn't even mention aside from in this clinical focus. So I wanted to hit prions just really briefly. They're infectious proteins. I'm going to come back to this picture. They are smaller than a virus even. They are just a protein, which a virus is either DNA or RNA wrapped in a protein coat. And this is more what a prion is. It's just a little protein. if that protein gets usually ingested into a person, i.e. you ate something that was contaminated, it can travel from the gut to brain tract and get into your brain and start to make the other proteins in your brain act inappropriately. Essentially, one prion can turn a good protein into another prion. So that's why they're infectious. One can make others bad. They work sort of like enzymes that way. One of the complicating factors for prions is you have naturally occurring proteins in your body that if you happen to inherit a genetic condition, you inherited a prion version of that protein instead of the normal version of that protein. So prions can either be infectious or they can be genetic. In the case of the infectious one, again, you usually ate something that was bad. Like there have been cases of Creutzfeldt-Jakob disease that happened because a person ate a cow that had mad cow disease. That cow had the prion. When you ate that cow, you consumed the prion, and then as a result, you ended up with Creutzfeldt-Jakob, which is what the gentleman here in this picture has, and you can see some of the very debilitating symptoms that come along with that. However, the case that they mention in this clinical focus is a person who has what's known as sporadic CJD. That means she ended up with a spontaneous mutation in one of her normal protein genes that turned her normal protein into a prion protein. So it's not because she ate something that contaminated her. She just happened to be exposed to a carcinogen that converted a normal gene into a bad gene that coded for a bad prion protein. So prions are complicated, essentially, is what it means. We'll talk about prions a little bit more later on, but they were mentioned in that clinical focus case, so I just kind of wanted to mention them. Hopefully that helps you understand more about the complicated stuff in Chapter 1.